think the clap was for Tom, not for you walking up, Kai. So, but I'm sure you'll get a lot of claps at the end.
Oh.
And so it's my great honor and to introduce my colleague, Professor Kai Voss-Fels from Geisenheim University, who's going to talk about some of his visioning ideas on developing predictive breeding capability for grapevine improvement. Over to you, Kai.
Yeah, thank you very much, Mark. Yeah, and thanks very much to the organizers. That's a great opportunity. Very special slot, like right at the end of this conference, to talk a bit about the work we do at Geisenheim University. So we are a small uni in Germany, but also an extremely specialized one. So we basically all the research and teaching we do focuses on production and processing of horticultural crops. Now, I've been very fortunate to be able to start developing a new department, the Department of Plant Breeding, with a young and very motivated team of people in which we're trying to develop strategies that could help to accelerate the crop genetic improvement process with a focus on horticultural species. So we've kicked off the session with Kirsten, who was talking about Arabidopsis as a model. Our model species is grapevine in this case, which turns out to be a really bad model for or model crop for developing predictive breeding strategies. And I'll be sharing some of the challenges we've been facing lately when trying to think about a bit hard of like developing predictive capability for breeding improvement. But I also want to share some of the excitement that we've sort of got when developing new data sets and starting to making progress in some critical areas. Now, grapevine in itself is I think arguably really it's a special crop. People or humans have grown grapevine since more than 8,000 years actually. So some say it's one of the most iconic crop in the world. So we've been producing it for a long time. But looking into the future, there's some pretty compelling or convincing forecasts that say that viticulture as a production system is particularly vulnerable to the impacts of climate change and such. Now, that obviously imposes a huge challenge on breeding and developing strategies to accelerate breeding or genetic improvement. And if we think about grapevine, there's a couple of really specific features associated with the production of grapevine, but those translate into the breeding of grapevine as well. The most important thing being that grapevine, as it's commercially grown in the field, actually consists of two genetically very distinct parts. So we got the scion, the upper part, which we use for grape production, for winemaking in our case. And we've got the lower part, the rootstock, that anchors the vine in the soil and sort of supports it with nutrients and water. And as you can imagine, with regards to breeding, the objectives or the breeding objectives differ very, very strongly depending on which part of the crop we're actually talking about. So we're trying to breed both or genetically improve both, the scion and the rootstocks, and then ideally also the combination, like breeding for the right combination of the two, which then creates a really interesting situation also with regards to G by G interaction or G by E or G by E by M. That's obviously, this can be interpreted in many different ways. Regardless of the part of the plant that we want to improve, there's a couple of things that make grapevine really challenging to work with. And I mean, could be listed a few more, but at least there's two things. One is that it has a really long juvenile phase, so it takes about 3 years until a vine produces grapes that you can actually measure. And obviously, quality is a super important trait, or trait complex, so it takes a long time. That translates into extremely long breeding cycles. I've worked with spring and winter cereals before. Breeders think 7 years is a long cycle, but obviously grapevine can easily go up into the decades. And the other thing being that grapevine is highly heterozygous, and it's a perennial crop, and it's being clonally propagated, which means that our capability of running nice MET sort of systems, multi-environment trials, is extremely highly limited. So those 2 points alone, I think, point out that there's a huge need, but also huge potential, huge leverage potentially, with regards to developing and implementing prediction modeling approaches at various levels. Now, I'll give a short example on some work we do on rootstocks. Now, rootstock breeding is actually, has been started around 120, 150 years ago almost, and it's typically done by hybridizing wild species of Vitis. Now, the concept of species in the genus of Vitis is a bit wild in itself, so you can quite easily intercross different species to create these hybrids, as depicted in this figure there. But then, and that's basically how we kick off a breeding cycle. And then we run through different stages of selection. And then if we're lucky, after about 30 or 35 years or so, we can release a new cultivar to the market, which is a very long time. Now, there's, again, becoming a bit more specific about rootstocks, it's extremely hard to phenotype rootstocks for commercially relevant traits. Now, one of the traits, for example, is drought tolerance that we're really interested in. But we generate thousands of seedlings, through crossing different species with each other. They go in the glasshouse, obviously, and then eventually we produce, or we bulk them up as cuttings, and then eventually, after a decade or so, they go into the field as single grafts in single fields, and then finally we can probably afford to send maybe a few dozen out to something that is comparable to a multi-environment trialing system as we know it maybe in cereals. So early stage MET testing is not possible, basically. That's just the situation. And we've got a lot of disconnectivity with regards to the environments in which we select in. So we need to sort out thousands of seedlings in the glasshouse, at least for a couple of years to start with. And then ultimately we need to select them for traits that have relevance in the field. So that's a big problem, connecting glasshouse versus field conditions. But then also connecting up different developmental stages. So we start off with seedlings, which we then bulk up as cuttings, like pure rootstock cuttings, and then eventually we graft them, we put them in the field. So that creates another level of disconnectivity. So we really do need accurate and most importantly scalable technology to sort of overcome, at least to some extent, some of these critical bottlenecks. And so I won't be talking a lot about genetics and genomics in this particular example, but I want to point out pieces of work that two really motivated and talented researchers in our group have been leading, Timo Strack and Carlos Robles. Unfortunately, this is the last talk. Everybody will run away after this, but they've been attending this meeting here as well. So in this example, on the left-hand side here, Timo was trying to sort of start developing proxies that we could use to screen rootstock populations for drought response. In this example, he was using the juice delta 13C sort of content or ratio. In this example, in segregating families of Vitis berlandieri, which is one of the most important species that we use for rootstock breeding. And that's actually quite elegant because it correlates quite nicely with the amount of stress a vine has experienced over the growing season. But most importantly as well is that it's very easy to measure, in fact, so we can measure it easily in juice, at least on plants that produce berries that we can extract juice from. So it's highly scalable, and it actually also correlates quite nicely across tissue types, so we can also extract information from wood tissue, for example, so that kind of ticks a couple of boxes for a nice trait that we can actually use to scale up our screening pipelines. Now, the other example, and this is work that Carlos has been leading, and he's tried out many different things in the last half year or so since he started in our group. And here you see some really interesting preliminary results from Carlos, in which he's used the hyperspectral sensor to sort of take spectral reflectance profiles on leaves of grafted vines in a very special trial type that we call adaptation trials. So in these trials, we fix the scion part, which means we use the exact same clone of a cultivar, which we then graft onto multiple different rootstocks. They're planted in the field. They've been planted like 15 or 20 years ago, so it's very representative of a commercial vineyard. And Carlos has collected hyperspectral reflectance data on leaves of Riesling, in this case, in 3 different developmental stages. And what's really impressive and kind of amazing to us is that we observe quite a strong genetic signal. Depending on the growth stage, we actually take those measurements, or Carlos takes those measurements, actually a lot of work, as depicted by the relatively high broad sense heritability estimates for the different wavelengths, which actually means we could potentially use remote sensing technology quite elegantly to extract interesting things like digital phenotypes, like growth indices, developmental indices, but also potentially use it for some sort of typing, phenotyping, genotyping, well, the phenomics equivalent of genotyping, which is actually quite exciting. So these are just two little snapshots with regards to rootstock improvement, which actually give us some hope that we might be able to sort of start building data sets that are at least big enough to start building predictive capability with regards to rootstock improvement. Now, the other example, and this is now focusing on the scion, is actually one that had, ever since I started this job in 21, has always fascinated me. This is about improving traditional cultivars via clonal selection. Now, viticulture is basically dominated by traditional cultivars. 95–97% of the viticultural area is planted with traditional cultivars, like Riesling, for example, in Germany, the most important grapevine cultivar there. And we've taken this year, actually this season, we've removed a couple of bunches from our diverse collection of Riesling clones and took this pretty photo. And it's actually quite amazing. So all these clones, all these bunches come from different clones of the same cultivar, that if you DNA fingerprint them with like microsatellite assays, for example, they're all Riesling, but they just look very different. And the reason for this is that we've been using these traditional cultivars for hundreds, if not thousands of years. So we had saw it on the bottles last night. We had I think Shiraz Tempranillo cuvée, very important traditional cultivars. Many people know Pinot Noir. Pinot Noir is over 2,000 years old. That's actually quite impressive. And we've been clonally propagating and maintaining it for a really long time, some cultivars being more than 5,000 years old, actually. And that has created huge amounts of diversity, actually, within these cultivars. And for me, as like someone who was trained and socialized with annual field crops, that's a concept that takes a while to get his head around. So there's variation within cultivars, extreme amount of variation, actually. And, well, growers to start with, but then breeders have been trying to systematically exploit that variation via clonal selection, and that looks pretty similar to a standard clonal selection program for a clonally propagated crop, with the main difference that we actually do not induce variation via recombination or via hybridization, via crossing, for example. But we, in our case, rely mainly on natural variation that we observe in the field, and then that we then assemble. We can also induce that artificially through mutagenesis protocols, and then basically the breeding pipeline and the subsequent selection stages just operate as usual with the timelines as indicated there. Now, this is a really interesting part of the work that we do in exploring clonal variation in some of the most important traditional cultivars. So this example here is from Riesling. There's a couple of people, including Hannah Robinson, Max Schmidt, and also Peter Crisp, a collaborator from the University of Queensland, who've been involved in this. So these clones, we have about 1,200 of them, have been planted in field trials for around 1.5 decades or so. And we've learned, I think, from Boris early on, it's more important to have more years than sites. So we only have one site, but we have very many years. So that at least creates some sort of G by E interaction. And what we actually see is there's huge amounts of variation with or across these 1,200 clones that are field tested. And this is just a snapshot of some of the traits we measure. We do measure more traits. We also have more cultivars, clones of other cultivars. And depending on the trait, and if we use some mixed modeling frameworks, we find that about heritability estimates, whatever heritability means in that context, but the genetic signal, as in this case indicated by broad sense heritability estimate, is actually pretty high. Now, simultaneously, we've started to investigate the amount of, or to look at these clones, but from a molecular perspective, and try to search and screen for both genetic and epigenetic variation. And this was done with Peter Crisp's group. So we've actually screened the 250 most diverse clones in this population. And this is also something that we've been scratching our heads around. We actually didn't believe that was true, but we had we find more than 1 million mutations across these 250 clones of the same cultivar. and almost 900,000 epi alleles, which are essentially differentially methylated CpG sites. So that was quite fascinating. Obviously, now we're trying to sort of bring this all together. And this is work that Hannah has just recently done, trying to sort of fit a couple of FA, or a couple of mixed models, in fact, in this case, an FA2 model, to sort of disentangle the contribution of the genetics versus the epigenetics on these complex trait variation patterns that we observe. And so effectively fitting a model with two FA terms, one taking in the genomic information or the genetic sort of variants to derive a relationship matrices, the other, the epigenomic information to derive an epigenomic relationship matrix. And so a large chunk or a large proportion of the variation can actually be assigned to genetics and epigenetics. It's almost like equal shares. And what's really interesting to see there as well, that the G by E, in our case, that would be G by year interaction, is playing out quite interestingly in the form that we see a lot of potentially scale G by interaction associated with the genetic term, and quite a bit more of crossover G by E associated with the sort of genome-wide differentially methylation or methylated patterns. All right, now there's a lot of work in progress. We're very active and very motivated, at least as we started out, to leverage proxy traits and use an array of different omics tools to build reference data sets and predictive modeling. That's very important. And we've become really interested in also addressing or using grapevine as a model species quite literally when it comes to sort of studying the interplay between genetics and epigenetics. And yeah, I think, the last statement I'd like to make is, I mean, I've really enjoyed interacting with all of you. I probably won't get the chance to talk to any of you because this meeting is over. So if you are interested in discussing this further, we're highly interested also in other crops like apple, for example, or started building or developing projects in vegetables as well. So yeah, please contact us. There's a big group of people, really excellent people. This is a photo of our team with the location in the background. It's sunny there sometimes, and we've got we're surrounded by grapevine, and we do have an experimental wine cellar. So yeah, come and visit us and reach out. Thank you very much.
Thank you, Kai. Got a very interesting laboratory also. Any specific questions?
I have a small question about your SNP data sets. Are those 1.1 million SNPs on only your scion germplasm?
Yeah.
So you didn't even look at the root parts?
No, exactly. So this was just leaf tissue.
Wow.
And DNA extracted
That's
from
nice.
leaves. Yeah.
Yeah, well, I actually also wanted to ask you about the SNP variation. Then if you have 1.2 million differences between your scions, at what point are you going to stop calling Riesling, and start calling it different varieties? So we have, as you can imagine, so we are in Germany, and we've got a very strict sort of IP protection system in place for breeding. So in fact, there were some clones depicted on this figure, or on this photo, that produce berries that are so small that a varietal authority like office in Germany would not accept them as Riesling anymore. Because there's about 3 dozen different traits. We call them registering traits or something, registration relevant traits. So Riesling is clearly defined as by the German national varietal authority. And if they deviate too much, even if you say, well, this is 99.999% Riesling genetically, then you wouldn't be allowed to register that as a clone of Riesling anymore. But the varieties are then registered phenotypically, not genetically at all.
I'm sorry?
The varieties are registered
phenotypically, yeah.
phenotypically.
On the other hand, you could breed something completely different if you manage to assemble the exact same trait configuration of Riesling. combine 30-something different traits, you could call this Riesling, even though it's genetically completely different. I hope the Bundesdatenamt is not listening to them. Oh, well, it's actually not possible, I think, but it's all recorded. No.
One thing I learned walking around the field is you should not ask that question. Causes a lot of controversy. Yeah. I'll take one last question and then we'll move on to Fred for the wrap-up session.
Hi, thanks for a nice presentation. Talking about these 1.1 million SNPs, can you say something about where they are in the genome?
So this, well, the genome is actually really small. So the haploid sort of set is only 450 meg big. And so it's extremely dense, like gene dense, which is also complicating some of our sort of genomics and bioinformatics. But they're clearly enriched for non-coding regions, because otherwise, I mean, they would cause a huge mess potentially. So we can clearly find that probably most of these mutations are in areas that are not directly translating into, you know, an imminent change in phenotype. That's what we can see that. That's probably the reason we find so many, because they not all of them do anything. Yeah.
Okay, thank you very much. Thank you very much, Kai.
Thanks.
Wonderful closing talk.